Hello, I'm Catherine Unsworth and I work within the Centre of Excellence for Long-acting Therapeutics, referred to as CELT, which is based in Liverpool. The work I will be discussing here is linked to one of the arms of the Longevity Project, on which CELT partners with Unitaid and several global collaborators, including Johns Hopkins University and the Medicines Patent Pool. Hepatitis C infection leads to significant mortality rates worldwide, mostly through liver cirrhosis and primary liver cancer. And unlike other strains of hepatitis, there's currently no vaccine against hepatitis C. As with other infectious diseases relevant to the Longevity Project, a disproportionate burden of hepatitis C lies with low and middle income countries. But you can see from this map that there are also high case numbers across the world, especially in North America and in Eastern and Mediterranean Europe. But the positive side is that hepatitis C is curable in most cases, with the use of combinations of direct-acting antiviral compounds which interact with the virus by more than one mechanism. Despite the availability of effective therapies for against hepatitis C, uptake can be low in certain regions and communities. Historically, there's been stigma around hepatitis C linked to perceptions of how infection is acquired, and subsequently there can be a reluctance for people to undergo testing. Additionally, infection is often asymptomatic and can sometimes clear spontaneously within the first few months. However, if it doesn't, it can sit undetected and transmission can continue over many years. Long treatment times and associated high costs are challenging for both treatment initiation and adherence. Long-acting therapies have demonstrated suitability for sustained release in antipsychotic therapy and for contraceptive purposes. And there are precedents for a long-acting therapy for hepatitis. An extended release form of combination pegylated interferon alpha and ribavirin has previously been administered as a weekly injectable therapy for the treatment of chronic infection. Glecaprevir and pibrentasvir, or GMP, form an effective direct-acting antiviral combination therapy. Both APIs are pangenotypic antiviral agents, which means they can be used to treat all major types of hepatitis C infection without the need for costly genetic testing. GMP are currently the only pangenotypic therapeutic option with a treatment time of less than 12 weeks and offer the opportunity for full curative treatment in as little as 8. These features make GMP suitable for the development of a single injection treatment, which may be used where access to healthcare facilities is limited. However, both APIs are characterized by low aqueous solubility, which makes the development of high concentration solutions or aqueous suspensions far from straightforward. One method of stabilizing low aqueous solubility APIs in an aqueous environment is by formulating them as solid drug nanoparticles, or SDNs. SDNs are particles that are made up primarily of the API itself and stabilized with polymers, surfactants, and sugars. They exist in the dry form, which increases their storage stability compared to liquid products, and these dried materials can be reconstituted in a suitable medium, for example water for injection or medical saline, prior to administration. SDN generation methods are versatile, and particles can be generated through top-down techniques such as milling and homogenization, or by bottom-up techniques including emulsion generation and particle precipitation. Particle properties, including particle size distribution and crystallinity, can be influenced through selection of stabilizing excipients and process solvents, and this can be used to target particle and powder material properties that are suited to specific administration routes. On the right-hand side here are a couple of examples of reported suitability of SDNs to the generation of high concentration aqueous dispersions of hydrophobic APIs suitable for injectable delivery. And these include the anti-malarial atovaquone and niclosamide, which is a compound with wide repurposing potential. Despite their low aqueous solubility, both GMP were found to be sufficiently soluble in several water immiscible solvents, making emulsion generation a viable processing option. Emulsion templated freeze drying was initially used to effectively screen combinations of stabilizing excipients and process solvents without the use of large amounts of API. Only a few milligrams were used for each candidate formulation. This is the screening process here. Half a mil emulsions were generated by sonication and then rapidly frozen in liquid nitrogen. The samples were then lyophilized to remove the liquid phases through sublimation, leaving a porous monolith which could then be reconstituted. Particle populations were characterized by DLS, and formulations that generated stable particle dispersions were considered for further development. Screening highlighted that both G and P could be effectively stabilized by PVP and docusate sodium, labeled here as AOT, and initially parallel formulations of each API were generated. I mentioned earlier that it was possible to adjust some of the particle and powder properties of SDNs through formulation and process parameters, and I wanted to show a couple of examples of this. On the left-hand side here are 3 overlain DLS traces representing SDNs of the same formulation, that is glecaprevir, stabilized by PVP and AOT, but prepared using 3 different solvents as the discontinuous phase. And you can see that 3 very different particle populations were generated. This narrow distribution here where dichloromethane was used, and more multimole populations where ethyl acetate and anisole were used. On the right-hand side are the results of microcalorimetry analysis of the same pair of stabilizing excipients, so that's PVP and AOT, and the calorimetry indicated some degree of interaction at the molar ratio represented in the emulsion, which may be indicative of the mechanism of API stabilization that these excipients provide. Going back to the parallel formulations of GMP that were selected from the emulsion templated freeze-dry screening, we can see from these DLS traces of monolith dispersed in water that the particle size characteristics are similar, with a slightly larger population of particles present in the pibrentasvir dispersion. From these SEM images, we see that the monoliths of both G and P, are amorphous in nature. And so at this point here, we've been able to demonstrate that it is possible to produce stable aqueous dispersions of these low solubility APIs. However, to understand if it's possible to develop high concentration aqueous dispersions suitable for delivering curative doses of these APIs, a method of generating much greater masses of material was required. And to do this, the emulsion templated freeze drying generation methodology was translated to an emulsion spray drying methodology. The principles are the same, but since the scale and the drying mechanisms are different, some slight modification to the process was required. Again, here's a brief overview. The API was dissolved in a water immiscible solvent, which was then added to an aqueous dispersion of stabilizing excipients. These were then sonicated using a probe sonicator to generate an emulsion, which was then fed into a spray dryer and dried using nitrogen to generate a fine free-flowing powder. And from this, 4 parallel formulations were optimized. Again, the single API parallel formulations. And since a key target of this program is to generate a single injection therapy, parallel co-formulated formulations were generated, one in which the ratio of G and P was 1:1, and one which is similar to the oral dosing option, at which G and P are present at a ratio of 2.5:1. The image here compares the correlograms and the particle size distributions of the 2 co-formulations, and we can see that they're fairly similar. The only notable difference is between these materials is the ratio of G to P, which is added by mass, and so may impact the molar ratios of APIs and excipients, but doesn't seem to impact the particle size characteristics, at least not at the relatively low concentrations at which DLS analysis was carried out. The SEM image here again shows an amorphous material, although with a more defined particle form as a result of spray drying than was generated by lyophilization. A key target in developing a long-acting injectable therapeutic option is to optimize the amount of API that could be injected in a single volume, as this will impact deliverable dose and potentially the extent of sustained release should we be able to demonstrate long-acting delivery further down the line. Intramuscular administration volumes are commonly up to around 2 millilitres and occasionally up to 5 millilitres. However, large injection volumes are reportedly linked to injection site discomfort. Additionally, high API loading may come at a cost to the ease of reconstitution of these materials and with higher injection force required upon administration. In addition to large injection volumes and injection force, the osmolality of an injectable product, that is a measure of dissolved material, has been linked to patient discomfort upon administration. Injectable products would ideally be around physiological osmolality levels of around 300 milliosms per kilogram, although levels of up to 600 milliosms per kilogram may be acceptable. In the present study, freezing point depression osmometry was used to measure these high concentration suspensions, with values of around 450 to 500 milliosms per kilogram determined. And some authors have suggested that osmometry of high concentration suspensions may vary depending on osmometric methods used, and so some further developmental work will be required here to fully understand how best to determine osmolality of these high concentration SDN dispersions. We wanted to know if we could anticipate steady release of these APIs in an aqueous setting, and this was carried out using a dialysis-based in vitro release study using a fast spin dialyzer and a size-selective membrane. G SDNs were dispersed at their maximum syringeable concentration and loaded into the dialyzer, which was then placed in water and allowed to agitate over a sampling window of several sampling windows of 2 to 3 days. The dialyzer was removed periodically and placed in a fresh vessel, and the amount of G was quantified by HPLC. And while we were able to demonstrate sustained release of G into water, this was nowhere near close to being suitable for representing physiological conditions. And so further in vivo studies would be needed to demonstrate long-acting delivery. The multidisciplinary nature of CELT enables preclinical assessment of formulated materials like these. So I hope that the studies presented here have demonstrated that it was possible to develop an emulsion spray dry generation methodology for candidate formulations of glecaprevir, pibrentasvir, and co-formulated G and P SDNs. It was also demonstrated here that characteristics of the formulation could be suitable for avoiding excessive injection site discomfort. Although in vitro release of G was reported, the studies presented here have not demonstrated the suitability for these formulations for long-acting delivery of G or P under physiological conditions, and this can only be demonstrated through in vivo studies by the CELT pharmacology teams. Some work that's currently being carried out by the CELT materials team includes assessment of storage stability and residual water, and we're working towards assessment of suitability for terminal sterilization by gamma irradiation. I'd like to take this opportunity to thank my colleagues in the materials and pharmacology CELT teams, as well as in other groups within the Department of Chemistry at the University of Liverpool. This work was supported by Unitaid as part of the Longevity project.